everyone. Today I'm going to talk to you about open biology, which is culmination of my experiences in academia. I've spent 13 years from starting from BSMS, then PhD, and also time as JRF. Uh, so in my talk, I'll be talking about the software, hardware, and the techniques and protocols that I have come across, which are open source and which are. So let's start with, can you tell me what comes to your mind when you hear the word biology? Anyone? Yes, life, cells, animals, planet, sorry, organs, yeah, mitochondria. Oh, that's nice. So you guys definitely know a lot deeper about biology. So these are just some of the words that I think people think when they talk about biology. And so this is where I want to concentrate on. The biology also refers to the money, patents, and publications. And it's often linked to health and life, and that is why it also is kind of related to the wealth that people make, and that is by having a lot of patents, because biology, most of the biological innovations, they are patented. And the biological research generally has three components. If you consider the pyramid structure, then the tip of the iceberg is the software. And in my talk, I'll be talking about these three aspects. Let's start with the software. So the most common software that I've used in statistics is R, because in ecology and evolution, like, you analyze and overanalyze and keep on analyzing data. And the paid counterpart for R is SPSS and Origin. So that is what mostly well-funded lab people use. For image analysis, I've used ImageJ, GIMP, and the paid counterparts are like Photoshop. For mapping the distribution of species, I've used QGIS, and the paid counterpart is ArcGIS. For referencing, I've used Mendeley, and the paid counterpart is EndNote. And one other very interesting software that I've used is from a spectrophotometer making company called Ocean Optics. So their software is Ocean View. So when you buy the spectrophotometer software, and you have to pay for the upgradation in the software. And if you don't want to pay, what they do is the older versions, they make it freely available, so that if you are not using the machine anymore, you don't want to pay for the upgrades, you can still analyze some of your data. So that was also very interesting aspect of making software free. And also, like, R also gives you that freedom to use these spectrophotometer data as well. So I didn't have to use this, I had to just use R. So going into a bit more detail, the SPSS actually roughly costs somewhere close to 79,000 per year, which is extremely costly. And R, on the other hand, is free. And the other very useful aspect of R, especially in academia, is somebody publishes a new paper, new technique, or a new method for analysis, and R will have it within months. But that's not the case with these paid softwares. It takes a lot of time for them to incorporate these new analysis into their regime. And some of the common packages that I've used is APE and tools for phylogenetic analysis, and for visualization, ggplot library and ggtree. So I'll give you a glimpse of like how we use these phylogenetic analysis and the scope of FOSS within this kind of analysis. Phylogeny is basically the history of evolution of species, and it's represented as a tree. You might have seen this kind of tree, right? Like how humans evolved and what their relationships are with primates. So that's what a phylogenetic tree. It's nothing but a hierarchical clustering representation with evolutionary relationships. How do you build such phylogenetic tree? You can build it based on DNA, morphology, or any other measurable feature, right? So DNA-based phylogenetic tree is the most accepted form of tree. And how do you do it? You have a selected set of genes, you extract the genes, you sequence them, It has 4 building blocks, ATGC, and then you sequence them, you align them, and then you run the phylogenetic analysis, and then you get the tree. So that's what phylogenetic analysis is. So is it only specific to ecology and evolution? No. So you can use this analysis for pathogen-host interactions, where you can track how the pathogens have moved from one species through the other species, so you can identify which are the susceptible species. And it can also be used for biodiversity assessment of microorganisms. Like in bioinformatics, the gut microbiota is a fat thing right now, right? So for that, when you want to analyze their relationships, what do? You build a phylogenetic tree and look at their relationships. So where do we need software and why do we need it? So firstly, to edit and align the sequences. And for that, sadly, we have only used Genius, the paid software, because there is no good software that allows you, freely available software that allows you to edit and align sequences. And when we are talking about aligning sequences, so we are basically talking about like hundreds of species and you're aligning sequences. So Genius is the only software which is paid. And if you are interested in biology and you feel like you need to contribute, then having a free software for Genius would be very good, because we spend like roughly 80,000 rupees per year to get the software. And to run the analysis, most softwares are free, so that's fine. And for visualization, ITOL and Figtree are the softwares that are paid, but you can do it very well with R. So that's the good thing. And in our lab, when I was doing PhD, we have completely relied on R. Apart from the statistics, we have also used imaging software like ImageJ to track the carpenter bee movement. And so basically what we do is we make the videos and then you convert them frame by frame and then mark bee in each frame and then look at their path. That's, again, because it's free software, it has been helpful. And then spectral clustering is one method that is very well known to the computer science people, but then when we were working on this biological system, we were not satisfied with the statistics that was currently available, so we collaborated with CS department and at ISC, and then we published this paper in an open source journal. And this is where my lack of understanding of FOSS comes in. Like, in academia, we don't know what FOSS is. So 13 years I've used open source software very heavily, but then I did not know how to contribute, and I just couldn't acknowledge, like I didn't know that it needs to be acknowledged. And we still have not made the code public, so it's still sitting in our GitHub repository, which we need to correct now. Now moving on to the open source hardware. So we wanted to build this pump for air sampling at a regulated time interval or in a regulated manner. So we constructed this flow pump, so you can see the versions, how it has evolved over time. The publication, all the data analysis, how well it works, is published in an open access journal, and all the construction details are there freely available on the GitHub if somebody wants to build it on their own. So let me give you a bit of background that why we had to go all the way to construct this. So this is what I had to do. I was interested in evolution of fragrances, why different flowers have different fragrances and how they have evolved. For that, I have to collect the fragrances. What we do is we send the air in at a regulated manner, and then we collect it at a regulated manner. So you need two flow pumps to collect one sample, right? And like most biological systems, we have a time limit. So since I was working with plants, flowering season for my plants is 3 months. And the number of species that I had to work with were 23. And I need at least 5 samples for each species, right? So that makes it like total 115 species. And I was working in the Northeast, so most of the days it would rain, so I cannot sample those days. So the number of days in which I had to do this sampling was even lesser. So for that, because each sample requires 2 pumps, so I need roughly 40 pumps, right? So the commercially available pump that when we inquired costs roughly around 97,000 rupees. If I need 20 units, then it would cost me around 20 lakhs, right? And what I need is 40 units, which is roughly close to 40 lakhs. And in ecology and evolution, generally, more than 10 lakhs grant is a very big deal. So having 40 lakhs just for a small instrument, which will keep going bad because you are out there in the rains, is impossible to imagine. So we built this flow pump where each unit costs 14,000, and so 40 units would cost me just 5 lakhs. So 5 lakhs versus 40 lakhs, right? So open hardware, and because it has helped us a lot, so we made it open source and we gave out all the codes, because in other countries, like a lot of people were approaching us for the codes, and so we had made it open source. So if they want to build it in their country, then they can do it. So other open source hardwares in biology is something called OpenPCR. So PCR is a machine. PCR stands for polymerase chain reaction, so which you use to amplify the genes, different genes. And the well-known brands, it costs somewhere close to like $5,000 to $20,000. But this OpenPCR machine is 100% free. If you want to build on your own, then the 3D graph, what is it called? 3D design, yeah. 3D design is freely available, and everything else around it is freely available. But then if you want to use their kit, which comes with all the box and everything, you can get their kit, which is just $500, so which is cheaper than an iPhone, right? Which again is very helpful. It's not completely free, but at the same time good. Then microscope. Microscope is another very essential aspect of biology. And there are these foldscopes which cost less than $1 to build. And they are freely available, I shouldn't say freely available, but anybody can build it. So their whole structure is available. And they are quite affordable. Now moving on to open techniques and protocols in biology. So in ecology and evolution, where I have worked mostly, so most of the techniques and protocols are free. So we don't have any patented technology as such in ecology and evolution. But then molecular biology people, they have tons and tons of techniques which are patented and which are closed source. So DNA isolation, gene transfer technology, gene expression technology, anything that you can think in terms of molecular biological experiment, starting from identification of the gene to identifying what function it does and how, if there is a problem with that gene, how do you fix it. So all these steps, every step requires you to have some dependence on either the software, hardware, or the techniques and the tools. But then they are not freely available. So when I was reading more about these techniques and protocols, like if there are any techniques or protocol which are out of patent or which are freely available, I didn't find any. But then I found this BIOS project, which is Biological Innovation for Open Society by Cambria Labs in Australia. So they follow a kind of a software model, like FOSS kind of model. So what they say is they allow you to get your techniques or like innovation patented, but with a clause that whatever new innovation you do, you have to give it back to the community free of cost, whatever the new development you do after using the technology. And it's free for the research institutes, like the academic institutes, and for non-profits. But if you are coming for profit, then when you request it, you have to pay some amount. So which seems like quite a nice idea, right? Like, you know, it doesn't have to be completely free, but at the same time, it's good. Then now, is it possible to have open biology, right? Like we are talking about the open source software, hardware, and techniques and protocols. So it's still difficult because incentives to not have your innovations patented is very less in India. Or I would say, I don't know of any incentive which the government or anybody gives you to not get your innovation patented. Right? And in today's time, like most of the faculties that I know in all these big government institutions, so there the government is kind of, the institution is forcing every faculty to have industry collaboration and get something or the other patented. So we are kind of like moving away from having open biology. We are moving towards like getting everything patented. Then comes the credit credibility and biosecurity. So if I am investing so much time, right? So, and I take some open source technology or like, some protocol, then how do I know that it's creditable, right? Like, because I don't want to use that protocol and waste my resources and my time. And then if I have developed something and I give it out, then how do I get a credit for it? Right? And what about the biosecurity? Now, once you make everything open source in biology, now anybody, because the genes and everything, those things, that kind of data is already open source. So if you have all that information, somebody can actually develop bioweapons. So I think for things like that, having a BIOS kind of system is good, because that way the technology is still in check, and people can still access it at affordable rates while also getting solving all these problems. And if you want to contribute to FOSS in biology, and also like towards making the dream of open biology true, then how can you contribute? So firstly, we need biologists, because they are the domain experts. So they need to identify what the problem is and find the right collaborator, because you can spend years and years, learning how to do machine learning for your problem, but then somebody in CS can do it in months, right? And then, so this whole idea comes mostly from, because I'm from academia and I did not know what FOSS is. So the students can, or from other departments, they can help by creating and maintaining FOSS for biology, create and maintain open hardware for biology, and the academia can promote interdisciplinary education, so that the biologists can also understand the language of a software developer or a hardware developer, right? So that is also important. And the academic course instructors can introduce FOSS to the students, and FOSS projects can actually be incorporated in the as a course project to the students. So that way, the students can contribute to FOSS. At the same time, they can also learn about how to, they can improve their CV, right? Yeah, so once broken will stop the wheel from turning, and that's what exactly happens in biology. So it has the innovations in biology has so many steps that even if one thing is missing, then you cannot move towards open biology. So open biology has to move like the software, hardware, and techniques and protocols together. Yeah, that's it. Thank you. So if you have question, I don't think I have much time. Maybe a minute. Okay, I can take 1 or 2 questions. Yes?
You mentioned the fragrance experiment you had done.
Sorry?
The experiment related to the fragrance you had done.
Yes, yes.
What was the outcome and what were the next steps? How did you follow it up?
We are still working in that field. So what we wanted to do is we wanted to look at the evolution of fragrances, and the hardware part was just one of the protocols. And what we found, like, it's a genus called Hedychium. I'm not sure if you're interested in it, but a genus called Hedychium, which is like a ginger lily. So what we found is that the species have day and night fragrance patterns. And the species, at daytime, they produce a different kind of fragrance, and nighttime, they produce a different kind of fragrance. And the species which evolved in India have different kind of fragrance pattern as compared to species that evolved in Southeast Asia. So yeah, so that kind of, and it's still an ongoing project, so we are yet to publish this whole work. Yeah. Yes?
Hello. Okay.
Yeah.
Yeah. I had a question. So you spoke about designing your own open hardware to like collect fragrance. Could you talk about what that device does?
So that device allows you to collect fragrance at a regulated, so it basically, when you make the chamber, you need to send the air at a regulated manner, right? Like, let's say 300 ml per second or per minute, you have to send in. And when you collect the fragrance, that also has to be collected at a regulated manner. So this device, what it allows you to do is it allows you to regulate the airflow. So we can measure like how much air is flowing out. So you attach a flow meter, which lets you know what is the flow meter, and then you can regulate it. You can set a timer for it, like you want to collect it for 1 hour, then it will beep. And it also has lightings, so that because we also collect it at night, right, fragrances, so it also allows you to locate the device at night because it has a faint red color light, yeah. Yeah. So analysis of the fragrances, we do it in GCMS machine, yeah. Okay, yeah, so I think my time is up. If you guys want to talk, then I'll be available today. So yeah, thank you everyone.